So just to kind of remind you, what we have, in my opinion, is we've got, let's see if I can get a laser pointer working, people or animals or whatever consuming antibiotics, and then we have people getting sick and or dying with resistant infections. And going from here to here, in general, sort of the 40,000 foot view is, I think, reasonably okay, but the details, I think, are lost. And the devil is in the details. So, well, I'm an academic. I'm flying down yesterday from Minnesota. By the way, I was really disappointed to learn that the temperature was going to be the same in Atlanta as it was in Minneapolis. I was looking forward to a little bit of warmer weather. And I was reading a paper, and it was actually, ironically, after the last presentation, on antibiotic use in agriculture, specifically poultry farms. And it was, I'll be honest, it was an awful paper. I seriously hope it doesn't get published. And the woman next to me, I have no idea what her background was, but she got really, really angry at me. Because what are you supposed to do about animals and this? And it was, yeah. So, in the spirit of antibiotic stewardship, I'm not going to talk about agriculture. I will tell you that when I got into wastewater treatment research, and I'll talk to you about my research in a little bit, I was very surprised that the wastewater treatment industry got very, very angry at me. Ironically, the next paper I read was a paper written by my friend Amy Pruden, and there was a really nice quote in there that I just, so this is, you might imagine that I was working on my slides on the flight here. That's not true, but I did add this slide. Antimicrobial resistance is a complex problem driven by many interconnected factors. Single isolated interventions have very little impact. So if you want to point a finger, you're going to need about 400 fingers. Okay? So just remember that. Anyway, I'm going to point a finger at wastewater treatment. Actually, I guess I'll summarize this. If you went to the wastewater treatment industry and said, are you ignorant or apathetic about antibiotic use and antibiotic resistance, they would say, I don't know and I don't care. So there's a real issue with how do you track antibiotic resistance in the environment. You could sort of take this medical approach, or there is the medical approach where you isolate a pathogen from patient, and then you sort of screen it for antibiotics, figure out which ones are effective, and then prescribe accordingly. I'm assuming. I'm not a real doctor. In the last presentation, you actually got a hint of sort of the model organism approach, whether it was Salmonella or Campylobacter. You target some model organism, you isolate it, and then you check to see which antibiotics are effective. Now, microbiologically, there's a problem with this in that we know that there are times in which organisms cannot be cultured, or we can't culture various other organisms. My postdoc advisor coined the phrase Great Plate Count Anomaly, and he hates that phrase, so I try to say it as often as I can in public. But it's still true. The method that we use is to not track organisms, but we track the genes that encode for resistance, and we do this by quantitative polymerase chain reaction, and it allows us really to get an estimate of resistance of a specific kind for a specific antibiotic. The trick here is there are large numbers of antibiotics and large numbers of resistance genes for each antibiotic. There are papers now in the literature where they are literally tracking as many as 300 genes simultaneously. We're not that good quite yet, but actually, to be honest, nor do I think it's necessary. Okay, so back to pointing fingers. This is a diagram that came from the CDC Antibiotic Resistance Threat Report 2013, and you will see that in the spirit of pointing fingers, there's the hospital side and there's the agricultural side. And I'll tell you this, I'm rather annoyed there's no municipal wastewater treatment side. Okay, so and I think I'll make the argument quite clearly that there should be. Now, this slide, okay, so one thing I tell my students, if there's any animation, I did not make the slide. So this is from my grad student, Tucker Birch, who now works for the USGS at a USDA lab in Marshfield, Wisconsin. So he was playing around with one specific gene, TET-W, which encodes resistance. It's a ribosomal protection protein for tetracyclines, and did some math with some data from the literature. And if you look at what leaves a wastewater treatment plant in the effluent, so this is the treated wastewater, it's on the order throughout the U.S., about 10 to the 17 copies of that a day. Now, who knows how right these are? When you looked at the sewage sludge, and I'll talk about that in a lot more detail, it's about 100-fold higher, 10 to the 19 copies per day. And when we looked at all of agriculture, what comes out of various animals being applied to farm fields, it's on the order of 10 to the 18 copies per day. The point of this isn't, am I right or wrong? The point is, municipal wastewater treatment is certainly on par with agriculture, okay, as far as importance, at least for this gene. So I will give you sort of my vision, which is a little bit different. The last talk was excellent. It's about antibiotic prevention, stewardship. I actually go the other route, and I think we should think about controlling antibiotic resistance. And we could do that simply if we think of, I have a stronger word that I don't use in public, but I call it my poo hypothesis. And that's the source of our resistance. And then I view wastewater treatment as a technology to get rid of that. Okay. So the research questions that we ask is, how effective are our current wastewater treatment operations? And then what could we do to make it better? Now, how many of you are wastewater treatment folks? One. Awesome. There's always one. So this is sort of just a general schematic of wastewater treatment. It's a simple process. You take raw sewage, you treat it with a bar rack. It's exactly what it sounds like. It's a rack of bars. A grit chamber to get rid of sand. A primary clarifier, which is just a quiescent settling chamber that lets things settle if they want to settle, let things float if they want to float. And you skim those things off. The particulates you settle off is organic rich, and it tends to go to some treatment process. We'll talk about that in a second. And then sort of the workhorse of wastewater treatment is an aeration tank. Basically, there's a lot of nutrients in wastewater. If you provide the right conditions, microbes want to eat those nutrients and grow. So that's what we do. In the secondary clarifier, we remove those microbes, send some of them back to the aeration tank, all the excess, because it's a growing living system. Again, tends to go to treatment. And then we send it out to the world, and maybe we disinfect. This part of the world, I'm guessing disinfection occurs year-round. In Minnesota, we don't go swimming in January, so we tend not to disinfect year-round. The bigger deals I'll talk about is what do you deal with these residual wastewater solids? That's the politically correct term that the wastewater treatment industry wants me to use. Everybody else calls it sludge. So what do you do with your wastewater sludge? There's a bunch of different technologies here, and then about half of it goes to landfills and about half of it goes to farmland. So there is an agricultural connection. So what is wastewater? Oh, I forgot one important thing. I will tell you, so here's sort of my outline of my talk. I'm not going to talk about what's in raw sewage. It's bad. I can promise you that. I will tell you this also, that I think some research needs to be done there. If you read papers in the literature, there's often these offhand comments. Well, of course there's antibiotic resistance in raw sewage. There's a hospital in that city. There's people in every city. Okay, so that's kind of a stupid thing to do. We've got a study that we haven't published yet, which is really ambiguous, where we tried to compare antibiotic resistance levels with medical activity, and we were unable to do so. I think basically this is the foundation of my poo hypothesis, which is it's bad. I tend to simplify things. So we're going to talk about what goes out in the wastewater. People really get excited about that type of research. That's what leads to hate mail to me. I will tell you it's probably not that important, not nearly as important as what happens at the bottom. There's two reasons for this. One, about 95% or more of the resistance that shows up in the raw sewage ends up down here rather than out here. And then the other thing, which is most important in my mind as an engineer, is we actually have a really good way of dealing with it, and that's that treatment technology that's right there. Okay, so this is my 1,000 words slide. Treated wastewater in general is actually pretty good from the perspective of an environmental scientist. Microbiologically, I have issues with it, but it looks really good. In general, if you look at the nutrient levels, it's on par with most surface waters. And in fact, in the Twin Cities, if you get overhead views of the wastewater treatment plants that discharge to the Minnesota River, you see this big black plume coming out of the wastewater treatment plant, and you think, oh, that's awful. And it turns out you're wrong. It's wonderful. the water coming out of the wastewater treatment facility is clear, and that black plume is the bottom of the river. You can't see the bottom of the river otherwise. So, it's good stuff. When you look at antibiotic resistance levels, and I should preface this, this is sort of log copies per milliliter of treated wastewater in the black dots, and this is 42 samples taken from wastewater treatment plants to discharge to the Mississippi River. I'll talk about the bigger data set later. And then the Mississippi River itself, and a bunch of different resistance genes. They tend to have about the same amount of bacterial biomass, so this is then total resistance genes. And what you see, and the numbers here give you sort of the information, treated wastewater is super rich in resistance, anywhere from, say, 100 to 1,000 times higher than what we see as far as background. There's a fun thing about Minnesota. It's a really nice laboratory. We have cities. We have a big city. I shouldn't say we have cities. We have one, although we call it Twin City. Essentially one metro region. But then lots of really pristine nature areas, and one really, really big lake. You've probably heard about the other 9,999, but Lake Superior is an exceptionally large, exceptionally eutrophic, exceptionally cold, and hardly developed lake. And this is the city of Duluth. And this is the St. Louis River. And what we decided to do was to look at resistance levels in the St. Louis River. And most of this is national forest, and even further upstream. And then into the Duluth-Superior Harbor, and then out into Lake Superior. And what our plan was, or at least in our mind, our model was pristine, 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 pristine. Oops, we're into Duluth, maybe dirty, and then back to pristine. Okay. And so we looked at resistance levels. I'm always pressing the wrong buttons here. So just in general, from left to right on all these plots, you're going from St. Louis River out into Lake Superior, upstream to downstream. And basically what you see is sort of a skewed bell curve in which there's a little bit maybe upstream, a huge spike, that's the wastewater treatment plant, and then you see sort of a decay out into the river. And so the one thing that I haven't told you is this is probably one of the best wastewater treatment plants in the world. They take what is required by law and then go another technology beyond that. Essentially, they were bullied into it because they discharged to Lake Superior. And Minnesotans have this idea that it's pristine and should be kept that way. So it's a very high functioning wastewater treatment plant. And so I would argue that even the best wastewater treatment plant, you can see that signature. That, of course, was sort of a short-term project, and I had a student who made the mistake of asking me, hey, can you advise me for a master's thesis? And I said, sure, let's go drive 3,000 miles. So back to Minnesota being my laboratory, we literally went up to the headwaters of the Mississippi River and collected samples. The letters are river sample locations, and we went all the way down to Iowa, and we did this 3 times. And then wastewater treatment discharges, which are in numbers. Now, the key thing is there's not a lot of people living in this part of the state. The Twin Cities is basically right here. And then this part of the state is very heavy with agriculture. And so what we did is we measured antibiotic resistance levels in all of these locations. And then we did something that engineers like to do. We did some math. So this next slide is super, super busy. And I've got a very simple point to make from it. The circles are the actual data, and then when we did the math, we've got a model in which we assume there's some level of background of antibiotic resistance, and so we start kind of here, and then we see the various wastewater discharges come in, and then it's diluted. The river is always getting bigger, and we assume it's diluted back with the level of sort of background level of antibiotic resistance. And again, so these are all different genes here, different dates. And then on the x-axis is sort of distance from the headwaters. And what we see with our model is in almost every case, we are over-predicting the resistance levels that we measure. Okay? What this is telling us is 2 things. First of all, there's a lot of decay in the river. Okay? Second of all, the river's really big. There's a huge dilution effect. The other thing I'll point out to you is you tend to see decay in the bottom part, or the far right part, the southern part of the state, which is actually where all the agricultural activity is. So, strangely enough, where we have lots of agricultural activity is where we measured the lowest levels of resistance in general in the state. So, that's the stuff that gets people excited, because for some reason people have more, I think, tangible attachment to water. They go fishing in rivers, they go swimming in whatever, so they see them. People tend to ignore the sewage sludge, and then as I've talked about, that's where the majority of the resistance shows up. In general, there's lots of different technologies. I would say the most common one is anaerobic digestion, which is basically to incubate the sewage sludge at body temperature and sort of decay the nutrients and get some methane out of it. We ended up, and this is lots of data, that's one of my grad students commented, you just said it went down. And that's really all I want to show you. These are lots of different technologies that we've gone through, and in all cases, resistance levels go down. And we can mathematically model those and then make predictions. This is what I think is the best technology, and this is a really busy slide, and I made it color-coded to make it a little bit more exciting. This is the effect of temperature on anaerobic digestion, and yellow is sort of ambient all the way up to blue, which is 55 degrees C, or about 130 degrees Fahrenheit. And what you see is as temperature goes up in these digesters, you get lower and lower levels of resistance level. And I think this is a really critical finding. Okay, and we'll come back to that in just a second. And then again, after there's treatment in the plant, it's often applied to agricultural soils, much like you would apply manure. I'm guessing many of you have used sewage sludge on farms or know of this, yes? Show of hands, maybe? Okay. And again, they go down, which is good. Now, what I didn't show you emphasizes the time scale here is always changing. So they all look like they're decaying at the same rate, but they're really not. And so the really important slide is this one, which is where we've computed half-lives for all these genes and all these different systems. And what you see is if you do sort of, and this is just 4 slides, if you wait to get rid of your resistance genes when you apply it to soil, the half-life is very, very slow. It's on the order of 1 to 2 months. Okay, if you do some very lazy technologies such as air drying or aerobic digestion, it's on the order of a week to 2 weeks. Certainly several days. If you use anaerobic digestion, thermophilic anaerobic digestion, it becomes something on the order of a few hours. What I'm saying is we can engineer the removal of resistance by choosing different technologies. Okay, so this is I think is pretty seminal. Okay, so I'm going to wrap up and just give you some take-home messages. First of all, we've got some new ways to quantify resistance that aren't sort of culturing these model organisms and looking at resistance with those organisms. All these methods have pros and cons. The nice thing about taking a model organism is you get to learn about that organism. Okay? The nice thing about studying the genes is we get around the cultivation bias. The problem with studying the genes is we don't know where they are. Okay? If they never hit a pathogen, we don't really care. Although we do know they get into pathogens. I will tell you that raw sewage is bad. You already knew that. I've often thought of titling this Everything You Need to Know About Antibiotic Resistance You Learned in Kindergarten. I just haven't. I will also tell you that treated wastewater, now again, the wastewater industry doesn't treat for resistance. They don't see that as a problem, and when I get hate mail, they're like, quit bringing this up. Okay, wastewater treatment are like, if you follow football, they're like the offensive linemen. You only talk about them when they're bad. Okay? And so they don't like to be talked about at all. Okay? The best I shouldn't say the best technology. The second best technology is thermophilic anaerobic digestion. There are some other people who disagree with this, but my research shows this, and thankfully somebody else is starting to show this too. We see very big improvements with removal of resistance genes when we elevate the temperatures. The best technology I will tell you is this, which is ironic because the folks in the Twin Cities do this and they stop talking to me. The best technology is incineration. You set it on fire. I haven't done the research yet, but I'm thinking all the resistance is gone. All of it, 100%. That tends to upset the air pollution people. But I'm just saying. So anyways, this is awesome. The people I've upset the most in the world are the folks in the Twin Cities and the folks in Duluth. Folks in the Twin Cities do incineration. I got this idea from the people in Duluth because that's what they do. And so I send what I think are love letters and they send me hate mail. I'm really insecure. Okay. So what am I really saying? Wouldn't it be awesome if we could collect a large fraction of the antibiotic resistance genetic material that's out there and destroy it? Okay? On the human side, we almost do that. We have this really wonderful wastewater treatment infrastructure, and we collect it all, we send it to one point, and we have treatment utilities there. They are not intentionally designed to get rid of antibiotic resistance, and they could be upgraded relatively inexpensively. Okay? So I will say this. My ideas are complementary to what everybody else is doing about antibiotic resistance. Most everybody, as I see it, is about resistance prevention. I'm talking about resistance control. I'm admitting that some level of resistance is going to exist. Let's hunt it down and let's get rid of it. Okay? So I see them as complementary. I'm sure some people will be like, hey, take all the antibiotics you want. Let's let wastewater treatment deal with it. I'm not a big fan of that. So with that, I will start acknowledging some of the funding. The Minnesota Environment and Natural Resources Trust Fund has been a wonderful supporter of my research through the years. They continue to do so. And a nice grant from the National Science Foundation. I couldn't have done this by myself. Lots of folks have contributed. Apparently Kyle Sandberg and Mike Sadowski